Hi, I wanted to talk about this worksheet. We did some practice on our autosomal pedigree with the Ed and Ethel and Mary and Joe. We had done this in class together last Thursday, I believe it was. And so we worked out the situation where in the dominant disease pedigree, we saw that you could have the dominant allele in both alleles and have the disease, and you could have just one of the dominant diseased alleles and be heterozygote and still have the disease. So we worked through how we knew that Harry was heterozygote, and we did that by looking really closely at Harry's children. Harry's wife is homozygous recessive, and Harry's children both have the disease and then don't have the disease. And in dominant disease, the only way to not have the disease is to have little a, little a. That means you got one little a from this parent and another little a from this parent, which with the homozygous dominant genotype, that would not be possible. So we cross that out, and we can make Lou and we can make Murray if Harry is heterozygous. We did the same thing with Murray and Betty because we could tell from the parents and from the children, you could see where the alleles would be required. We didn't have any puzzles like that for this side because luckily and unusually, we actually know all the carriers. And there were a lot of carriers in this family. Therefore, it was quite surprising that Lucy and Fred and Bill and Marge and Ethel and Ed never actually got the disorder, but Marcia sure did. So Marcia has the homozygous recessive genotype, and that gives her, PKU disorder. We then went through and answered these questions. These questions are going to be due by all students on Wednesday at the beginning of class. So make certain that you're working on this little by little if you didn't get it done in class today. The next thing we did was we took a look at how to answer a genetics question when using a Punnett square. We made certain that everybody understood a Punnett square is just a tool that we can use to determine all the possible offspring. These are the gametes from one parent and the gametes from another parent, and we have to split them. Remember, sex cells are haploid, so that means that they only put half of the alleles into their respective cells that they're producing. So if mom was big A, she would only put one of those A's into her egg cells, and then the other dominant A allele would be in the other half of her egg cells. If we're talking about dad and he's heterozygous, then half of the sperm cells he makes will have a capital letter A on the genes that are in those cells, and then the other half of sperm that dad would make would have the recessive allele or the recessive A allele on those particular genes. You put them together, if this sperm and this egg fertilize each other, that might be the offspring. If this sperm instead fertilized this egg, then this would be the offspring or the zygote. If this sperm and this egg came together, it would look very similar to the one above it, but that just is the way this all pans out. And then if this sperm and this egg come together, that would be your possible offspring. So this is looking at probability of outcomes. A Punnett square is done with each and every pregnancy. Just because you might have these two children doesn't mean that you can't have these with a third child or a fourth or a 10th child. It's always all of these options with each new pregnancy or with each new fertilization. Then we went down the page a little bit and made certain that we understood how in the world to answer a question when someone is asking us a genetics question, and it probably will be asked or probably will require us to do a Punnett square. They're going to ask us very specifically, tell me the genotypes of the offspring, or tell me the phenotypic genotypic ratio, or maybe just tell me the phenotypes of the offspring. Or they'll be very precise and say, what's the phenotypic ratio of the offspring? So you have to be careful the way that you answer the question. Some of the key pieces are you have to address every one of the genotypes or every one of the phenotypes, and you have to go in order from dominant to less dominant. So as you move across from left to right, you're going to start with the most dominant presentation. In this case, A up here is one of, these are Mendel's pea plants, and these are some of the traits that he tracked and made very famous. Axial flowers are branches that will have flowers on either side, and a terminal flower is a branch that will only have a flower at the end of the actual branch. So he thought that axial flowers were dominant when both axial parents and terminal parents were bred. Almost all of the offspring were axial. So therefore, he said axial must be dominant, which is a little true and a little misleading, but he went way beyond what I'm mentioning. So we're going to take a look at this. We crossed a heterozygote parent with a homozygous dominant parent, and these are the offspring we got. So the genotypes of the offspring, we just list. We got two big bigs and two big littles. If they ask for the genotypic ratio, then we're going to say we got two big bigs, because that's the largest or most dominant genotype, compared to, or ratio, two big littles, got those over here, and then we have to include the one genotype we didn't get any of. So we say 0, little, or homozygous recessive. You can write a short version, so 2:2:0, but you've got to remember not to lose that placeholder 0. When they ask about phenotypes, that's the physical expression of the trait. So what does it look like? How is it expressed? You're just going to give a list. So 4 axial flowering plants are in this offspring box. Every single one of those offspring will have axial flowers. And then the phenotypic ratio. So that means you've got to refer to both the axial and the terminal. Start with the dominant, that's axial. So you'll say 4 axial compared to 0 terminal. Again, if you want to do the short version, that's a 4:0 ratio. So then we practiced. These pieces are our crosses. This is a heterozygous, heterozygous for smooth pod shape, crossed with a homozygous dominant for smooth pod shape. So you can put this one on the top or you can put this one on the side. It doesn't matter where you place them, but you put one parent here, cross all the options. Again, big, 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 come across. We want to try and make certain that we get all the possible combinations. And you see there's not a lot of variation. You either have homozygous dominant or heterozygous presentation. So genotypic ratio, go from most dominant to least. So 2 homozygous dominant compared to 2 heterozygous compared to 0 homozygous recessive, or the short form, 2:2:0. Phenotypic ratio, again, what does it look like? So 4 smooth compared to 0 constricted. None of those offspring are constricted. And again, the shorthand is 4:0. Now you try. I'll give you these with the crosses written out. When you go to the other side, you'll actually have to start by writing the cross, and then you can do the Punnett square. But there is a cross and a Punnett square required in addition to all the answers. Thank you. And most students were able to get this done in class, but not everybody. And it is totally okay to take this home and work on it there, but it is due tomorrow. Thanks very much and have a great night.